All right, we're going to continue with cells, and we're going to spend most of our time now talking about the eukaryotic cell, the true cell, because there's a lot more stuff to a true cell than there is to a prokaryotic cell. Okay, when we look at a eukaryotic cell, we have a lot of genetic information. This is the information that is passed on as the cell divides, as this organism reproduces, and so on. So the genetic information is housed in the nucleus, and its work is to make proteins, and that's actually carried out by the ribosomes. And we talked about that a little bit with transcription and translation, and we'll talk more about that later. But the DNA is the one that has the blueprint to build you. All the information about you, your eye color, your hair color, your height, the fact that you have 2 legs and 2 arms and 2 eyes and 1 nose, that's all in your DNA. Okay, that's where it all comes from. So that's in the nucleus. And normally you can't see it. When a cell is ready to divide, you can. If you've ever seen a cell with the little X's in it, the chromosomes, then you can see it. But normally you can't see it because it kind of spreads all out. Okay. The ribosomes use the information from the DNA to make proteins. Okay. So the DNA is the information. It's copied by RNA, and then it goes out to the ribosomes, and that's where the work is actually done to make the proteins. Okay. So a nucleus contains the cell's genes. Again, it's DNA. And usually it's the most conspicuous organelle. In other words, if you look at a cell, you can see the plasma membrane, you can see the cytoplasm, and you can see the nucleus. Most of the times you can't see anything else, they're just too small. But the nucleus is the biggest of all the organelles. Okay? And it does have a nuclear envelope. It has its own special membrane. Because again, we have stuff that has to stay in the nucleus. We do not want our DNA escaping the nucleus. We need it to stay in there so that it has this envelope that surrounds it to make sure that what's in the nucleus that's supposed to stay in there stays in there. It also kind of works like the plasma membrane and allows some stuff to go out. If messenger RNA copies the DNA and needs to go out and make a protein, it's got to leave. So again, it works kind of like a security gate. Some things can come in, some things can go out, some things have to stay in, and some things can't come in. And that works in the nucleus as well. Okay, it's a double membrane with the bilayer, like I showed you with the regular. So it's very, very similar to the outside layer of the cell, the plasma membrane, this membrane that goes around the nucleus. And again, it's called the nuclear membrane. Basically, it works the same way in allowing some things to leave and some things to come in, but some things can't leave and some things can't come in. Okay, in the nucleus, DNA is organized into units called chromosomes. Okay, chromosomes look like little X's. And when a cell divides, those chromosomes are split up evenly, and each new cell gets the same amount. And every living organism has a certain amount of chromosomes. Humans, we have 46. Okay, so we have 23 pairs of chromosomes. Okay, each chromosome is composed of a single DNA molecule. And then there's proteins called chromatid that kind of hold it all together. And we'll talk more about that when we talk about the cell dividing. But that's what's inside your nucleus. Chromatid is the protein that holds DNA together. When you see a chromosome, and you can see this under a microscope when a cell is ready to divide, they look like little X's. They are DNA and chromatid protein. That's what a chromosome is. And that's all the information to build you. Okay? They condense when the cell is ready to divide. Instead of this long, it'd be like taking a long piece of string when it's all stretched out and then coiling it down so it's only about this big, instead of, when it was stretched out before, maybe it was 5 feet long, but it was a single strand. Now you coil it up, and that's what a chromosome is. It's coiled up DNA. Okay, and then there's the nucleolus. It looks like a little dot in the nucleus, and it helps make ribosomes. Okay, there's another RNA, ribosomal RNA, that helps in the process. It kind of anchors the messenger RNA down. And so there's a little spot in the nucleus that helps make that. And it's called the nucleolus. And under an electron microscope, it would be inside the nucleus, you'd see another big kind of round dot. That would be the nucleolus. Okay. The ribosomes are the little organelles where the protein is made. They look like little dots. And they're ribosomal RNA and proteins. And what happens is they're the site of where the protein is actually made. If you watch the video on translation, you see the ribosomes, and They're the factory. A ribosome is like the factory where the proteins are made, okay? And they carry it out in two different places. Okay, the ribosomes carry out protein synthesis. They do it, sometimes they have free-floating ribosomes. And they also have ones that are attached to what they call the endoplasmic reticulum. How's that for a big word? Okay, the endoplasmic reticulum is a layer of it's hard to describe. It goes back and forth. It's like a membrane, but it's back and forth, and it's always by the nucleus. Okay, and there's two kinds. There's smooth and there's rough. If it's rough, it means it has ribosomes attached to it. If it's smooth, it doesn't. Okay. Another thing that they say the endoplasmic reticulum does is it helps me move the fluid. It's like a mixer to keep things moving around in the cell. Okay, so the endoplasmic reticulum, and you can just say ER, not emergency room, you guys. It accounts for about half the total membrane in there. It's a huge organelle. Well, it's not as big as a nucleus, but it's always by the nucleus, and it's just basically a bunch of big membranes. Okay, it's continuous with the nuclear envelope, so it's kind of connected to the nucleus, and it can have ribosomes on it, or it can be what they call smooth. It doesn't have ribosomes. Okay, so if its surfaces, you'll see little dots on it. That's rough endoplasmic reticulum, and that's where you're going to have some proteins being made. If it's smooth, it doesn't have ribosomes, and they actually have two distinct regions. You can see it on a cell. Okay, so the smooth ER, endoplasmic reticulum, helps make lipids. If you remember that, lipids are fats. Okay, and it helps break down, metabolizes carbohydrates. In other words, they come in as complex and we break them down into smaller parts. Because remember, we really need to use glucose. Glucose is a monosaccharide. You put a bunch of monosaccharides together and you have a polysaccharide, which is a carbohydrate. So we need to break that down and use it. Okay. A smooth ER helps get rid of poisons and drugs, especially in your liver cells. Okay, that's their job, is to take care of, to detoxify when you take in things that you're not supposed to take in. Okay, it helps store calcium, which we need for our bones and for nerve function, all those things. So smooth ER does a lot of things. And again, it helps move materials around in the cell so it can grab hold of stuff and send it where it needs to go. Okay, rough ER, the one with the ribosomes, okay, they secrete something called glycoproteins. They are proteins that are actually bonded to a carbohydrate. So they can help it escape. Because remember, when you looked at the plasma membrane, it had proteins that they'll attack to there and they'll help get those carbohydrates out. So if the cell is producing carbohydrates in it, let's say it's making glycogen. We talked about glycogen is the way we store energy. Well, what if that glycogen needs to be stored in your muscles and your liver, but it was made somewhere else? Now its job, maybe it was made in the intestines. Its job is to take those carbohydrates and get them out of the cell and bring them back, bring them to the muscles of the liver where they need to go. What are we doing time-wise? Oh, we got lots of time. Okay, so they are transport vesicles, they're called. Okay, their job is to take whatever was made in the cell, bring it to the membrane, and help pass through. Okay, so they transport it. They're like security detail. They'll get them to the gate, and then they'll help them pass through security, and then they'll go out into the bloodstream, and then the blood will take it where it needs to go. Okay, and it's the membrane factory for the cell too. If you need more membranes, that's where they're made, in the ER. So the ER has a lot of jobs to do. Okay, the Golgi or the Golgi. Again, I was always told Golgi, but I've heard Golgi by a lot of people, so it's up to you. The Golgi apparatus is like the UPS of the cell. Things come in there and they make sure it gets to where it's supposed to go. So they're the shipping and receiving center. Okay, it's flattened cells. I'll show you a picture, but there's like 3 flattened squished cells, and then they have little dots on each side. Okay, they're called cristern. And what the Golgi apparatus or the Golgi apparatus does is it takes the products of the endoplasmic reticulum and it packages them up and modifies them so that they can actually be sent out to where they need to go. Okay? It makes some other macromolecules too, but again, really what it does is it receives stuff from the cell and it modifies them enough so that they can be shipped to where they needed to go. All right, so again, the Golgi apparatus is like the UPS center. Things come in there, they package it up, and they send it off to where it needs to go. Okay, so it sorts and packages the materials into these transport vesicles, which are usually as a type of protein. Okay, so again, Golgi apparatus is like the UPS store. Okay, then there's lysosomes. Lysosomes are little packages of digestive enzymes. Okay, and their job is if there's stuff in the cell that needs to be gotten rid of, it's waste, it's junk, they'll go up and they'll secrete some of their enzymes and break it down. Okay, so the lysosome's job is to get rid of waste in the cell. They're little packages of enzymes. Okay, they work best in acidic environments inside the lysosomes. So their enzymes are really good at helping break down whatever's in the cell that's not supposed to be there. Okay, so you always end up with some waste products. They need to be gotten rid of, and that's what the lysosome does. Okay, there's hydrolytic enzymes. Hydrolytic meaning they have water, and then the membranes, the hydrolytic enzymes and the lysosome membranes, they're made by the endoplasmic reticulum. They transfer them to the UPS store, the Golgi apparatus, and then they process them until you get these lysosomes. They also called lysosomes, I've heard them called the suicide sacs, because if they released all their enzymes at one time instead of a little bit at a time, the cell would die. That's from what I've seen or I read about was like, a lizard, when you grab its tail and the tail comes off and the lizard runs away, there's actually like kind of like a ring of cells there. And what'll happen is they get that stimulus of being pulled and they'll release all their enzymes from the lysosomes and all those cells will die. And that's why the tail comes off, it detaches, and then the lizard can get away. So whoever's trying to eat it only gets a tail and the lizard lives to see another day. That's an example where the lysosomes would come in handy, and that's when they're the suicide sacs, but really losing their tail is saving their life. Okay. There are some cells in there that can engulf food particles. That's called phagocytosis. This happens again. There's certain one-celled organisms like amoebas. That's what they do. They surround something and they make a little food vacuole, and then it floats around and the food goes wherever it's needed. The lysosomes can fuse to that food vacuole and digest the molecules, the stuff that's not supposed to be, because when you break down food, there's stuff you need and stuff you don't need. The lysosomes get rid of the stuff you don't need. Okay. And they can use to recycle stuff. If they have a macromolecule there and there's something in there that's big, they'll go down and break it down into smaller parts so it can be reused. So their lysosomes always also recycle components inside the cell. Okay, the process of breaking those big molecules down is called autophagy. Phago means to eat. Okay, so they're eating, stuff inside themselves or whatever. So lysosomes really are like the digestive and the excretory system. They get rid of a lot of the waste. Okay, there's vacuoles in the cell. Vacuoles are very large vesicles. They come from the ER and the Golgi, and what they do is it depends on, you have two different types. You have food vacuoles and you have water vacuoles. Water vacuole will hold excess water and then it'll take it to the membrane and get rid of it so the cell doesn't explode. Okay. Let's see where we at. Food vacuoles are formed from eating big parts. Phagocytosis, again, they chomp it. Okay. And then there's contractile vacuoles. That's what I was saying. These are the ones that you'll find in freshwater animals, ones that live in the water. You got to get rid of some of that excess water inside the cells. So the contractile vacuole holds the water, kind of goes to the membrane and like squeezes it out so it doesn't explode. It doesn't, too much on the inside and then the membranes can't happen. And there's also central vacuoles, which you only find in plant cells that'll hold organic compounds. And we'll talk more about them, I think. Yeah, we're just about the end here. We'll leave it right here and then we'll talk about these again to review it. So that's it for today. Again, don't forget to do your assignment.